In this video we're going to talk about DNA replication. That's something that happens inside of a cell in the nucleus before we can do either mitosis or meiosis. So that's the context of DNA replication, sometimes called DNA duplication, but they mean the same thing. So let's go delve into the nucleus and look at the DNA itself. Here I have an example of it. This is just a very small portion of what is a huge amount of DNA. At this particular scale, this DNA would probably be the length of a football field if we were talking about an entire chromosome. So we can't do that whole thing. We'll just use this small snippet to see how replication works. The key to replication is partly its simplicity. There are 4 different nucleotides that make up DNA, and it knows how to make the new strand by matching complementary nucleotides. So complementary to adenine is thymine, complementary to guanine is cytosine, and cytosine obviously to guanine. So a fairly simple formula. If this is one strand, this is what the other strand looks like. So DNA serves as its own template. So we need a couple of enzymes at work here. One is helicase, and the other one is DNA polymerase. All enzymes are specialized tools. They do one job and they do it really well. Helicase has an interesting job. This little guy comes along with his big teeth and he unzips and unwinds the DNA. Remember that these here represent hydrogen bonds between the nucleotides, which are fairly weak, and so he can move along and unzip the DNA fairly easily. Now that he's done that, our other specialized tool, the DNA polymerase, can come in and start laying down nucleotides. But it has to do this in a very specific way. Let's only back up for a minute and note that DNA has a very specific way it's put together. One end is what we call the 3' end, the other one is the 5' end. In other words, there's a beginning and there's an end. If we went this direction, we'd be reading it backwards from how DNA polymerase works. So the DNA polymerase, it's a very specialized tool, but it only knows how to operate from the 3' end to the 5' end on this old strand. Can't read backwards. And since DNA is anti-parallel, I might note that this end over here is the 3' end. This is the 5'. And so that end over there is the 5' end. So DNA polymerase would have to work this direction, which is forwards, even though it's right to left to you, on this strand of DNA. So I'm just going to take this one. I'm going to lay down a work surface for this. Now normally these nucleotides would be floating around free in the nucleus, and DNA polymerase comes in to act. Let me bring this down so you can see it better. And it's going to match up a guanine with that cytosine. It's going to match up a thymine with that adenine. And when it does that, it's going to make sure that these have a covalent bond holding them together. So with thymine, there's adenine, and again a covalent bond. With adenine, there's going to be another thymine and another covalent bond. These are going to form these hydrogen bonds between these two as they go along, and that keeps them together. So DNA polymerase just keeps coming along and working guanine, cytosine, cytosine, guanine, adenine, thymine. After a while, it's kind of a boring job. This enzyme works very rapidly and efficiently, but I've used this strand of DNA as a template for making the new strand. I didn't even need to refer to the old strand, but you should note that this new strand in purple is identical to the strand below it in terms of the letter sequence, even though it's maybe written a little sloppier. Same sequence, so I just remade this strand using this strand as a template, using just the simple rule of complementary base pairs. So now I have one double-stranded piece of DNA. Incidentally, This over here would be the 5' end of the new strand, and this down here is the 3' end, so it's anti-parallel again. And the same thing is going to happen here. We're going to work backwards, but rather than walk through all that again, I've just produced this one. And so now we have two pieces of DNA. They're both double-stranded, and these would be sister chromatids. The cell would keep those stuck together and go into meiosis or mitosis after that. So that is the process of DNA replication, which comes before mitosis or meiosis and is mainly ruled by complementary base pairing using our handy enzyme, DNA polymerase.